Well, good morning. This is Dr. Miller. As you know, I'm in Cincinnati this morning attending the Society of Sensory Professional meetings. And I hope all of you did well on the exam. I know some of you are getting ready to take the exam right now, or while I'm gone. This lecture is only going to be about 40 minutes. We are very honored to have Miss Nicole Culley from JBS and Pilgrim's Pride, who's going to come in and talk to you at the end of class. I also want to remind you that you do not need to come to class on Friday, that the lecture will be recorded and will be online, as well as this lecture. And so you can watch the lecture at your leisure. I will be back on Monday. But I'd like you to be at class today on Wednesday because this is a unique opportunity for you to hear about JBS and Pilgrim's Pride and the opportunities they offer. Those of you who are planning on attending RMC this summer in Fort Collins will have the opportunity to work with JBS as they are a co-sponsor of the RMC this year, and they are a wonderful company. Well, we had finished talking about exsanguination. We talked about how this is blood removal. I gave you the story, just to help you to remember, last Friday about the ostrich slaughter and really trying to get full blood removal. And we know that exsanguination is very important. Well, the remaining steps, last slide on this, remember we're looking at the 40,000 foot level here. The remaining physical steps for converting a carcass or an animal into a carcass is after exsanguination, we're going to remove the feet, the hide, the head, and the tail. And you can see from these pictures that the hide has been removed. It starts to look like a carcass. We also have complete removal, except for the kidneys, of the viscera and the plunk. You had 307. You know what happens as far as removal of the byproducts, which become part of the dressing percent. That is very, very important. During this process, we'll have washing, we'll have application of interventions, and of course, immediate refrigeration. As we talked before, this process, which used to take about 22 minutes, now takes about 45 minutes. So that the physical conversion of an animal to a carcass is a fairly rapid process. But what I want to look at now is what's really going on down at the skeletal muscle fiber level. And we know, you've seen this slide before, we talked about this in the energy for metabolism section of the class. And we talked about how this is all a biological system that there's checks and balances, homeostasis, right? And we have the delivery of nutrients, the removal of waste products, the ability to replenish the system and to keep it balanced. Well, what we have just done during the conversion of that animal to a carcass, at the point of exsanguination, we have cut off this supply line where we can no longer bring in nutrients from the blood system, which mainly includes oxygen. Also, we can bring glucose in. So oxygen and glucose are the two main items that are brought into the muscle so that it can keep working. That's its food supply. But we've also cut off right here the venous supply to remove waste products. So we can no longer remove lactic acid, CO2, water, or heat. So that's a very critical step as well, because those byproducts, they can't get out. So what happens during this process is, kind of again, that 40-foot view is we're going to convert this muscle that's going to be able to move. This fiber is going to be generating aerobic metabolism. And then as we shut off the supply lines, both for food and removal of waste products, we're going to start having a limitation of oxygen. What's interesting is that glucose usually doesn't become the rate limiting component because there's still glycogen left in a muscle after conversion of muscle to meat. But we also have the buildup of lactic acid, of CO2, of water, and of heat. So during this process, which we call rigor mortis, which stands for the Latin definition of stiffness of death, and it occurs soon after death, and it's characterized by stiffness and extensibility of the muscle. Basically what this is the conversion of the muscle to full contraction, and it loses its ability to relax. That is, as that process occurs, as the muscle loses the ability for aerobic metabolism to function, it decreases the ability of this muscle to produce ATP. If there isn't enough ATP, eventually we're not going to have any ATP on those myosin heads, and they can't drop off. Remember the little yellow star of ATP that swooped in and attached to the myosin head, and then the myosin head could unhinge and release? We get to the point where we don't have ATP, so those myosin heads become permanently attached. And during the process of rigor mortis, we have almost 100% of myosin heads attached to actin. In normal contraction, we have about 10 to 20% of the myosin heads going up. And remember on the CD-ROM where the myosin heads were going up and it kind of looked like it was like some were going up but some weren't, and they went up at different times? That's what happens during normal contraction. But during the process of rigor mortis, those myosin heads will eventually just get stuck because there's no ATP. And the muscle loses its ability to produce ATP. So this muscle is trying to maintain homeostasis. So as this muscle, which has been stranded out there, no more nutrients coming in, no ability to remove byproducts, that which causes circulatory failure, we know that we're going to eventually have oxygen used up. Remember, myoglobin, which binds oxygen, only binds a little bit. And that's going to be the rate limiting step. That decrease in oxygen to slow down the ability of the mitochondria to work. What is that? That's conversion of aerobic to anaerobic metabolism. And we're going to have buildup of nutrients, mainly lactic acid, and we're going to have heat buildup as well. CO2 and water don't really play that big of a role in this system because water and CO2, they're produced in small amounts. We can't get them out. Not good, but it's the lactic acid that's going to cause some changes. And then we're also going to have some heat that causes heat, if you remember, will cause protein denaturation. So basically, again, just reiterating the same point, this is the conversion from an aerobic to an anaerobic environment. So in the living tissue, again, somewhat of a review, but I think it's important. I put these on the slides. As you know, I don't like to put writing on slides, but because I'm giving this lecture to you orally and I'm not here in person, I felt it was important to do this. So let's look at what's going to happen in the living animal when we are converting from aerobic to anaerobic metabolism. We know that oxygen, which is picked up by the lungs, is carried to the muscle by the hemoglobin pigment, and myoglobin sitting there. It's 1/4 the size of hemoglobin, and it's sitting in the muscle. It's what makes it red, and it has a greater affinity for oxygen than hemoglobin. So it grabs the oxygen from the blood and stores that oxygen until it's needed. So the amount of myoglobin and the efficiency of the myoglobin is what dictates how much oxygen is going to be there. We already know that with different muscle fiber types, some muscle fibers have different levels of myoglobin. So in this living animal muscle fiber that's just gone through exsanguination, there's not going to be any new oxygen, so oxygen's going to become depleted. And as that oxygen's depleted, it slowly becomes rate limiting and slows down the ability of the mitochondria to function. Because we know oxygen is needed to bind to the carbon when we take in the TCA cycle, when we're taking pyruvate apart. And we also need oxygen in the cytochrome chain to bind to hydrogen to produce water. So if we don't have oxygen, those two things can't happen. And as the mitochondria slowly shuts down, that energy metabolism gets shifted to that anaerobic pathway, which means that as the pyruvate and the 4 hydrogens build up in the sarcoplasm, lactate dehydrogenase says, oh no, and it puts pyruvate plus hydrogen and gives us lactic acid. But lactic acid cannot get out. And as lactic acid builds up in that muscle fiber, in the sarcoplasm, pH starts to drop. As pH starts to drop, there's a lot of things that happen. So as far as postmortem changes, that depletion of oxygen that reverts the aerobic energy metabolism to anaerobic causes the buildup of lactic acid. So we can monitor how rigor is going by monitoring how much lactic acid is there. Well, that's not that difficult of a chemical process, but really the buildup of lactic acid is parallels the change in pH. So pH, that postmortem decline in pH, which is an indication of how much lactic acid we have, is the most significant event in muscle. So as meat scientists, we monitor rigor mortis, the process of rigor mortis, by monitoring pH. And when you're seeing pH drop from 7.4 down to 5.6, which is the normal pH drop in normal muscle, you're going, oh, aerobic metabolism is converting to anaerobic metabolism, and lactic acid is building up, and pH is dropping. So that's why we monitor pH. Now, the rate and extent of that pH drop is very important. And in lab, you're doing pork quality, you're going to talk a lot about that rate and extent of pH drop. And you're going to taste some products to see if it really does make or affect the eating quality of the product. But depending on how much glycogen is present, that can affect pH drop. And temperature can also affect pH drop. If we can't get carcasses into the cooler in sufficient time, when it's 105 on the slaughter floor, the process of conversion of aerobic to anaerobic goes much, much faster, and we see pH drops go much more rapidly. And so temperature is very important, and it's important we get that heat out of the muscle as rapidly as possible, because temperature affects how rapidly biochemical reactions occur. The postmortem changes are also dependent on ATP concentration. If you have a well-rusted muscle, there's lots of ATP. Obviously, the muscle doesn't have to work as hard at the beginning to keep ATP concentrations high. And you can see this in pH decline. So temperature is important, and also we're going to talk about calcium concentration. The sarcoplasm can affect this, but mainly the biggest point I want to get through to you on this slide is that pH, which is inversely related to lactic acid concentration is, as it increases, pH drops. And therefore we can use pH as an indication of how rigor is proceeding. This slide shows you that pH drop, which I think you've seen before. And we know pH decline goes from 7.4 to about 5.6 to 5.7. It can go lower, it can stay higher. Those are abnormal pH drops. pH drops can go rapidly, or they can go at a normal rate, or they can just barely go at all. So when we look at this chart here, we see that there are different pH drops, and each of those pH drops are an indication that something is going wrong. Notice the green line here is a normal pH drop. That's what we want within the muscle. The muscle is going to start about 7.4, and we're going to see pH slowly decline. It depends on species, but in pork it can happen in 15 minutes to 3 hours. In poultry it's going to go much more rapidly. In beef and lamb, with electrical stimulation, which we talked about that in the blood pit with exsanguination. We may also use that prior to exiting the slaughter floor. It's going to drive pH to be a little bit more rapid. Now we have two situations here where we have much darker meat or slightly dark meat, and those are situations where we don't have a normal pH drop. We don't even get down to normal pH. What's happened there is we've used up some of the glycogen. Here we've used up almost all the glycogen with pre-slaughter stress, because the animals are using glycogen as energy production. So anytime those animals are having to utilize ATP, either because they're excited and they're moving around, they're not relaxed, or they're bad weather, can also do those type of things. We'll talk about that more later in the semester. Or we can have animals that may not be affected as much, so they still have some glycogen. So that initial glycogen level, as long as it's normal, we're going to be at the green line or below in pH drop. We only see these conditions or conditions in between that when we've had pre-slaughter stress that's long-term and the animals used up their glycogen, and that's the rate limiting step. But for the normal versus the pH drops that are below it, if we have conditions where either we have very high temperature or we have short-term pre-slaughter stress, so the animals are stressed very right as they're going into to immobilization, or something happens out in the pens as they're moving that way, that we call short-term pre-slaughter stress, something that happens fairly soon before immobilization, usually up to 15 minutes, 45 minutes, an hour. But what happens with those animals is we have stimulated the fight or flight mechanism. They've sped up metabolism. So we have normal amount of glycogen, but the animal is in fight or flight mode. So we've revved up the engine, and then we take the brake off because everything is wound up, metabolism is wound up. So aerobic to anaerobic metabolism, that conversion is going to go very rapidly, and we get a very rapid pH decline. We can get versions of that pH decline in between the bottom orange level with this kind of chartreuse green and purple line, where we don't have quite as severe of a pH drop. So below normal, that's due to short-term stress and speeding up metabolism. Above normal is due to long-term stress, where the animals used up their glycogen. What we would desire as meat scientists is that the animal would go through the process of rigor mortis and have a normal pH decline, because then we have normal color. We have been able to keep temperature from building up in the muscle too rapidly and causing protein denaturation. And we're also going to have some normal, we'll have water holding capacity, which will be normal. We'll talk more about those when we talk about pork quality. But I want you to know that as we're going through this process of rigor mortis, and we know pH is dropping because we're building up lactic acid, because we've just gone from aerobic to anaerobic metabolism because of the limitation of oxygen. There are a lot of things that are going to fall apart in this muscle fiber cell. With blood removal, we know we've lost temperature control and the circulatory system. And that rise in post-mortem muscle temperature that we see immediately post-mortem affects membranes and membrane fluidity. Think about the sarcoplasmic reticulum, especially the terminal cisternae of the sarcoplasmic reticulum. Remember, it has a lipid bilayer. If temperature goes up too high, that membrane starts to leak calcium, and because it's more fluid, that calcium can bind to troponin and we get contraction more rapidly. Lots of things going. And so we see here, I've kind of outlined some issues that will occur with increased temperature, or things that will be affected by temperature. If you have a larger muscle and it is located deep in the thickest part of the carcass, you're going to have a higher temperature. We know that along the femur bone and the round, which is the thickest part of the carcass, the inside of the outside round, the inside of the inside round, are the place and the carcass where we have the slowest temperature decline, and it's going to hold in heat. And in those locations, if you have a chilling problem with your carcasses, then what happens is you'll see more discoloration in those cuts. The amount of fat that's covering the muscle is going to also affect whether temperature and heat can dissipate more easily. Fatter carcass, as you know, fat has an insulatory effect on temperature that heat can't transfer through fat as rapidly. So if you have a really fat carcass, it's going to chill less rapidly and it's going to hold the heat in. The ambient temperature in the slaughter area has an effect on how rapidly carcasses are going to decline in temperature. The main reason for that is that they're going to spend 45 minutes in that ambient temperature. And I told you I grew up in Colorado. So in the wintertime, sometimes the slaughter floor is the same temperature as the cooler. In the summertime, that slaughter floor can get up to 100 degrees. So that obviously has to affect how rapidly that carcass is going to chill and how we're going to be able to get heat out of there. Length of the slaughter operation also affects temperature decline. We've already talked about this. It's always an issue if you have a chain breakdown on the slaughter floor and carcasses are held longer. carcasses that are pulled out of the rail for further inspection. And they may sit there for 10 minutes, 15 minutes, 20 minutes until the issue's resolved. Those carcasses are going to have a slower or more rapid temperature decline, and rigor is going to go more rapidly. And temperature in a chill cooler, as well as whether there's water chilling and what the air velocity in the cooler is, has a really big effect. What most plants do is the first cooler they put carcasses in, which is called the chill cooler, almost always has a spray system because water is a good medium to carry temperature, to keep the carcasses, get them colder faster. It has very high air velocity because it's trying to really circulate that cold air, and it's going to run very close to 32 degrees, if not slightly colder, depending on if the maintenance people can keep the lines from freezing up. It's the coldest place in the plant, and you want every carcass to chill the same way. It's very difficult in large coolers where you are putting carcasses on lines and you're packing them in there. You want to have really good air circulation. And then after 24 hours, those carcasses are usually moved to the second, or what we call the grading cooler, waiting to be processed for grading, where they'll be there 12 hours maybe, depends on the plant. Why do we worry about temperature? Because obviously it affects how rapidly pH decline is going to occur. It has a very direct effect on the pH declines from the previous slide. And when we see a low ultimate pH in the mussel, and you think about the inside part, the meat along the femur, the thickest part of the round, it's going to have a higher temperature, and you're going to end up with a lower pH, ultimate pH, and that's going to affect protein solubility. Protein solubility is really important for processed meats, and if the proteins can't function correctly, like we expect them to function with converted proteins that are in meat, then we're going to have problems with our further processed product. Because protein solubility impacts water holding capacity and the ability of the protein to bind to each other. We also lose the intensity of the mussel's pigment, and that happens where we see that usually a lighter color or a change in color. So lower pH is usually lighter in color. You're going to learn that in lab if you haven't already been in lab. And a higher pH is a darker color, and we'll talk more about that next week. So that's really what's going on, but I want to kind of put this together in a little bit different way. I want to look at how rigor proceeds, and we define rigor by 4 phases. We know that the formation of the permanent cross bridges in the muscle between actin and myosin is what makes it stiff. We know that there are the same chemical reactions in living muscle and the process of rigor mortis. And basically we talked about that relaxation cannot occur because of lack of ATP production, so that the myosin heads can't drop off. But we also don't have enough ATP during the onset of rigor or during rigor to complex with magnesium. We can't slide things back even if we could get the myosin heads down. So as ATP is depleted, we get those permanent cross bridges to form, and this reaction is irreversible without ATP. But we've lost all the mechanisms to produce ATP as we build up lactic acid, because pH is dropping. And all the enzymes involved in producing ATP are very pH sensitive. Well, let's put this into a little bit different thought process. There are 4 phases of rigor mortis. And you need to know these. There's the delay phase, the onset phase, completion phase, and resolution. This teal line here shows the extensibility of the muscle. And it's really the extensibility of the muscle that we use to define these 4 phases. And notice on the vertical axis, the measurement there is extensibility. So low on this axis is elastic and high is stiff. So during the delay phase, this is a short phase where the muscle's very extensible. It's very much like living muscle. It really hasn't had anything that's become rate limiting. But during onset, the second phase of rigor mortis, we start to lose extensibility slowly, and onset, that phase ends when we have 100% of the cross bridges formed and everything's stuck. Then we start the third phase, which is called completion. And resolution, another name for resolution is meat aging in the cooler. But we see that we gain some extensibility with resolution because we're going to have some loosening of the structure. That's due to protein degradation, not the actin myosin heads changing, but only degradation. So what I'd like to do is start with each of these phases and look at what's happening. Well, in the delay phase, the first thing I want you to see is that creatine phosphate in the red dashed and dotted line at the top of the delay phase. Notice that creatine phosphate is declining. During the delay phase, the first short-term source of ATP is creatine phosphate. Now, I'm going to give you a little hint. Creatine phosphate is gone in the delay phase. No more creatine phosphate. So you notice that right below creatine phosphate is the concentration of ATP in the green dotted lines. So ATP concentration that's needed for the myosin heads, the calcium pump, the sodium potassium pump, the magnesium ATP complex, those 4 things, which was a question on the exam, right? That ATP, this muscle is able to keep ATP concentration high. But it's using up creatine phosphate. It's starting to use up some of the glycogen, isn't it? Glycogen is going to be fed into glycolysis and then into the mitochondria because there's still oxygen. And we're going to be able to produce those 37 ATPs from aerobic metabolism. So ATP is able to be maintained, and glycogen is going to start to decline, and the rate of glycolysis is going to be fine. It's going to be able to still function. But what we find is that during this process, glycolysis is fine, but the mitochondria are using up the oxygen that's present. Remember, oxygen is going to be limited now. It's only what was stored in the muscle through from the blood system through myoglobin. So oxygen starts to decline because it's being used up. The mitochondria function, which is the red square dashed line, the mitochondria is still able to function because it still has oxygen. Everything's just trying to survive here. But then onset starts. And I've added one thing here. I've added pH decline. And notice that during delay phase, we don't see a pH decline because we have aerobic metabolism occurring. There's no lactic acid because the pyruvate's going into the mitochondria. Mitochondria is functioning fine. But during onset, we start to see pH decline. And that decline is inversely related to the loss of extensibility. So the loss of extensibility is related to myosin heads not being able to release. So basically going into full contraction. But look what's happening. Creatine phosphate and I went ahead and just put this all on the line. It's done. We don't have any more creatine phosphate to worry about. These are all relative concentrations. But look what's happening with ATP. ATP concentration is declining, somewhat parallel to pH. Isn't that interesting? That means that ATP, not being able to maintain ATP concentrations, we're converting from aerobic to anaerobic metabolism, and we're having a buildup of lactic acid. I added lactic acid to this to onset. Notice it was parallel in the delay phase and very low, and we see the concentration of lactic acid in that teal dash line increasing inversely related to pH decline, and it's parallel to loss of extensibility, because that lactic acid is what's lowering the pH, and it's what's causing the mitochondria. Obviously, it's an indication that the mitochondria have shut down or slowly shutting down. They don't shut down at once, they just slowly lose their ability to keep working, and we get that lactic acid buildup, and ATP is not able to be produced at the same level. Well, we are going to continue to use up glycogen slowly. We use it up a little bit faster in delay than on onset, because we're still having glycolysis go right here. The rate of glycolysis, it starts out where it's functioning really well, but as pH declines, those 12 enzymes involved in glycolysis slowly lose some of their ability to work, because they're maximally active at physiological pHs. So as we lower pH, it slows down their ability to continue to work. So we notice that rate of glycolysis declines, and we hit kind of a point where it says, oh, I'm really struggling. This point of inflection right here is where it starts to really slow down. And notice the oxygen. Oxygen continues to decline, and we get to a point where we pretty much shut the mitochondria down, and we're solely dependent then on glycolysis for ATP. At this point, that muscle cell is really struggling. It's trying to maintain homeostasis, but pH is increasing, can't get rid of lactic acid. It's really struggling. And look at the mitochondrial function. It slowly loses mitochondrial function. So there's no more ATP to be produced. It's in full contraction and has no ability to relax. In completion, nothing changes. Everything's parallel because everything's complete. In resolution, we gain some extensibility, and we also see a slight decline in pH. This decline is due to microorganism growth. Microorganisms that produce lactic acid and other acids, which are spoilage organisms, we will see help to decrease pH. But everything else is pretty steady. It's done. It was done in completion. It's done in resolution. We know that we're going to see a little bit of lactic acid increase again. That's related to lactic acid produced from microbial growth. And we'll see that slow degradation of some of the structural proteins. I want you to know the 4 phases and be able to recreate this slide. I do it slowly with the 4 slides, because if I just started here, I think I'd overwhelm you. The textbook shows this, and you're free to read this. I think that this is on this is figure 5.1 on page 115, and I think if you read it after you've studied the graphs, the last 4 slides, and listen to this lecture, it's going to make more sense. But it's just another way of putting the process of rigor together, similar to what I did, but in a different form. So that's the end. I think now you should understand how mechanisms of homeostasis induce physiological changes in postmortem, and also understand the steps of rigor and the physiological changes in the muscle that result from rigor. I'm going to stop now and say thank you to Nicole for coming into class and talking to you. Have a good Wednesday, and I'll see you on Friday morning. Goodbye.